a new genetic combination, two nuclei brought together. As a species that reproduces sexually, we all start life as a single cell, and it's unique. It's a recombinant of our parents' genomes. The zygote and all of your somatic, which are your body cells, as opposed to the gametes, the sperm and ova, the somatic cells are diploid. We give that number 2n, 2 sets of chromosomes, or 46 chromosomes in a human. Whereas our gametes over here, they need to halve their chromosome number first, becoming haploid, or just n. This is so that when they combine, the diploid number is restored, and each generation has the same number of chromosomes. In spermatogenesis and oogenesis, the germline cells, they replicate via mitosis, but the daughter cells that eventually go on to become spermatocytes and oocytes carry out meiosis. Although in either case, a cell can't divide without first making copies of its DNA, so DNA replication takes place. Here we can see one chromosome, and then it gets duplicated, and it's still called one chromosome. It simply has 2 copies of every gene, and each side is called a chromatid. Where that fits in the cell cycle, if we have a look over here, we have a series of growth cycles, G1 and G2, and between that we have S for synthesis, and that's where the DNA replication takes place, and our chromosomes get duplicated. For mitotic cells, there's one division, and the stages of nuclear division include prophase, metaphase, anaphase, and telophase. Might come over here for a minute, draw a cell and have a look how that changes throughout those stages. And in this example, I have not drawn all 46 chromosomes that a human would have. It's just a simplified cell with 4. My skills with the stylus are just not that good. So we have our cell here, tragic spaghetti-looking chromosomes. Now, what we've got here, I'm showing you, this is a cell before the mitotic division has happened, and now this second cell I'm showing you is prophase. This is the stage where the chromosomes condense, they get shorter and fatter, and we can see them under a light microscope. The nuclear envelope starts to break down, and we have these centrioles moving to the poles of the cell, and they're going to create those spindle fibers. We can see that now in the next picture we have metaphase, where the cells are lining up at the equator or middle of the cell. And where the spindle fibers attach is the same place that the 2 duplicated arms, if you like, of the chromosome are held together at the centromere. So this is the centromere there drawn in red. That's holding the 2 pieces together, the 2 chromatids. But it's also where kinetochores assemble, and the spindle fibers, they're little microtubules, and they can attach, which is what's going to pull them apart to either pole. And if I draw that next bit there, okay, so this here is what we call anaphase, where one of my past students decided a good mnemonic would be to call it anaphase because Anna farted and the chromosomes left the room. So they have split their centromeres at this point, and the spindle fibers are attached to either one, and they will pull them to the poles or either ends of that cell. And we can see it's elongating as well, because this cell is getting ready to physically divide too. And what we can say as well, when these chromosomes, when they have split those centromeres, what we called 2 chromatids when they were together, now that they're separate, each one gets called a chromosome in its own right. So we would say the chromosomes are moving to the poles. And then we finish here with telophase. So they start to relax, those chromosomes, the nuclear envelopes start to reform. And then hand in hand with telophase, we have cytokinesis, the physical division of the cell, the splitting of the cytoplasm. So that's not actually part of the nuclear division. I'm just going to pop over here just to reinforce, by the way, what I said before, that one chromosome is made of 2 chromatids when they're joined together. And those spindle fibers joining at the centromere, just pointing that out, and just emphasizing a couple of those things. Like I've highlighted there, the nuclear envelope breaking down, the centrioles, if you missed that before, moving apart anaphase, telophase, nuclear envelopes reforming. And that cytokinesis again, that is the division of the cytoplasm. And that little dotted line just showing for a moment there, not to dwell on it, but if you were in a plant cell, you'd also need a cell plate to form because they need to have that cellulose, that cell wall to be built to. All right, so that's mitosis. So we said those somatic cells are from the zygote. So most of the cells in your body, that's how they arise. They come from mitosis. But if we want to make gametes, we need to go to meiosis. And that's actually got two divisions. So let's have a look at the first one, meiosis 1. And the 1 goes along with being primary spermatocytes and oocytes. They're the cells that are involved. The big difference with meiosis, essentially we've got a lot of the same physical processes happening as in mitosis, but the big difference here is that chromosomes don't line up in single file. In the first division, they actually get together in pairs. So in these cells, and if you didn't know already, you've got 2 copies of each autosome. And an autosome, they're your chromosomes except the sex chromosomes, except the X and Y or some combination. So all of the other chromosomes are called autosomes, and they have the same size. The ones that are pairs have the same size and structure as each other, which means we can use the word homologous to describe them. So homologous means something that's the same structure. You might have heard the word homologous used talking about evolution as well when we talk about features and structures, sort of anatomical features of animals, like the pentadactyl limb with the arm, say of a human, the front leg of a horse, the wing of a bat, because they all have the same bone structure underneath. So we would call them homologous. And with our chromosomes, we can say they're homologous because they match where their centromeres are, and actually it's because they have the same gene sequence on them. Now, most biological females have 2 X chromosomes, and most biological males have an X and a Y. And the X and Y are structurally different to each other. They actually carry different genes. So we wouldn't describe them as a homologous pair. And here I'm attempting to draw a rough karyogram of a human. I'm not really sure why I didn't just cut and paste one because that would have made so much more sense. But I'm drawing one with an X and Y. So this is a karyogram and it's very rough. Technically speaking, these chromosomes should be arranged in decreasing size order, but I did not start big enough. The homologous chromosomes and the sex chromosomes, they do come together in pairs, so the X and Y can still pair for meiosis though. And they come together as these pairs during prophase 1. And they stay in those pairs when they line up in metaphase 1. All right, so this idea of a homologous pair, let's take one here and kind of zoom in a bit, I suppose, if we can. Now, a pair, these homologous pairs, they carry the same gene sequence on them, but the alleles, so the different versions of these genes, might not be the same. But the position where we find a particular gene on a chromosome is called the locus. So the locus is kind of like, the address of the gene here, or loci for plural. So here, looking at this one, another piece of terminology we can use, we can refer to this as a tetrad, and tetra comes from meaning 4 of something, and that's because there are 4 chromatids, or 4 pieces of DNA with those genes. And remember, they come together here. This is happening in prophase 1. And that's where the homologous pairs come together. And at that point, when they actually come together, and they do physically join as well, we can refer to them, this tetrad, we can also call it a bivalent. And here we can see the bivalent has 2 chromosomes. That's where the bi comes from. And each one of those chromosomes is comprised of 2 chromatids. And the chromatids that are part of the same chromosome, they're called sister chromatids, and they should have the same alleles. They should actually have the same versions of the genes, with the exception of perhaps 1 or 2 changes due to random mutation during that process of DNA replication. Where we have chromatids they're still within the homologous pair, but if they come from the 2 different chromosomes, we would call them non-sister chromatids. It's more likely that they will carry different alleles. So in prophase 1, the non-sister chromatids, they can cross over. That forms a chiasma. That's a singular term, or chiasmata in plural. That's the crossing point. and they physically break, and that occurs at the same loci, the same position on each chromosome, and then an exchange occurs. So this crossing over gives us, well, it holds our bivalent together actually, and it can occur in several places. I've only shown one chiasma here, but 3 or so are kind of typical for, especially for our larger homologous pairs. And the shuffling of alleles as a result of this means some of the chromatids are recombinants. And we used that word earlier to say that a zygote was a recombinant of its parents. And the word recombinant just means like any new combination of genetic material. So in this case, the chromatid that's been crossed over, it will be pulled apart to make a new chromosome with a new combination. And so we can say that's a recombinant too. We can talk about a whole zygote being a recombinant organism, or we can talk about a chromatid here making, and once it breaks away, to form a recombinant chromosome. So let's say we have genes A and B on these chromosomes, and that this person in our karyogram here, looking at this example, let's say they're heterozygous, so one of each allele for both genes A and B. So we'll put dominant A and B on this one and recessive A and B on this one, just to have a look at how they get switched over because of the crossing over of the non-sister chromatids. You can also use a horizontal line notation to show this. So it's a bit like the chromosomes have been tipped over sideways. And we can see how, we start with our 2 different chromosomes, the red and the purple ones. That's what we're trying to represent. And the chromosomes get duplicated. We talk about a chromosome duplicating because the DNA gets replicated. And then the crossing over occurs, and the non-sister chromatids, they have their exchange fragments, and so those ones in the middle there become the recombinants. And they're called recombinants because the combinations didn't exist before. So back to the process of meiosis and fitting that in with the rest of the picture. Actually, in oogenesis, the production of ova or eggs, that stops here at prophase 1. So we don't get very far. The process doesn't continue until a primary follicle is stimulated in readiness for ovulation. So when we say a biological female is born with all her eggs, technically she's actually got all of her primary oocytes instead. Whereas spermatogenesis, the process to produce sperm, that's continuous. Once puberty begins, the primary spermatocytes, they continue through this process. So after prophase 1, they go straight to metaphase 1. Got metaphase, so they're meeting in the middle, and this time they're meeting in pairs. So our bivalents are actually lining up along the equator. And then one chromosome from each homologous pair is pulled to a pole in anaphase 1. And after grouping at the poles in telophase and the physical division of cytokinesis, so that's the same as before with mitosis, what we've ended up with is we have 2 cells, and each one of those cells after telophase 1, each one of those actually has half the number of chromosomes compared to the parent. So the first division, meiosis 1, results in haploid cells, n number of chromosomes. But each chromosome is still duplicated. It still has 2 chromatids. So we still have a larger quantity of DNA than what we need. And that can be a little bit confusing because you think, you haven't got to the final point of cutting down the quantity, I suppose, of your DNA, or if you were looking at focusing on the chromatids instead. But in terms of the actual chromosome number, you have halved it after that first division. So by the time you get to secondary spermatocytes and oocytes, they are already haploid. Okay, so our second division, this is just like mitosis again as well, and in this case they do line up in single file. So we have the centrioles repositioning. We go 90-degree angle typically from the first division, and actually we have new spindle fibers forming here in the beginning of prophase 2, and actually a secondary oocyte stops here too. So we said before that when a girl is born with her eggs, really they're actually primary oocytes that are stopped at prophase 1. But then if one goes on to mature for ovulation, even at ovulation, that egg that's released technically isn't an egg. It stops here at prophase 2, and it will only continue through the next steps if it's fertilized. So they get suspended at several points. So the next bit then, we've got our single file lining up in metaphase 2. Then the centromere is split like in mitosis and our chromatids are pulled apart. Although actually I should point out that this isn't always successful. If the chromatids don't separate here in anaphase 2, or even if a whole bivalent stays together in anaphase 1, the distribution of chromosomes in the gametes won't be equal. And we call this failure to separate non-disjunction. So that's where we get an unequal distribution. Either the centromeres don't split or the bivalent doesn't separate properly. So it can occur in meiosis 1 or meiosis 2 that you get this happening. And one result of non-disjunction is trisomy 21, or 3 copies of chromosome 21 in a person, which is also called Down syndrome, or monosomy X, where only one X chromosome is present out of the sex chromosomes, and that's called Turner syndrome. And there's a couple of others as well. And there's some evidence to suggest that some of the connections between sister chromatids deteriorate over time, which makes the prevalence of non-disjunction increase with maternal age. That just means like the kind of faulty separation of those chromosomes is more likely to happen in eggs of older mothers. And because they've been sitting there, they've been stuck there for a while. But that mechanism, more research is needed to be clear about that. But if we continue as in most cases, I'm just going to use a more typical example where we don't have non-disjunction. The chromatids would separate just like mitosis. So they separate here, we call it anaphase 2. And of course, they're no longer chromatids. Once they're apart, they are called chromosomes, and off they go to the poles. And we have new nuclear envelopes form, and cytokinesis follows in telophase 2. What this results in then, from our original single parent cell, we have 4 haploid daughter cells. And the term daughter has nothing to do with whether they have X or Y chromosomes. It's just the terminology we use to say that they came from the parent cell. So whether they're sperm or eggs, they're all daughter cells. And actually in sperm, the division of cytoplasm and organelles is pretty even. All 4 gametes are usually functional, and they mature from being spermatids to having flagella, the tails and the acrosome and things, and the midpiece with lots of mitochondria there. And that's when they're called spermatozoa. With eggs, it's a bit of a different story. The ova, they have to be a lot larger because they're going to contain the bulk of the cytoplasm and organelles for the new zygote after fertilization has happened. So the division of cytokinesis is quite uneven, and the remaining small cells are called polar bodies instead. So we only get one ovum from a parent cell in oogenesis. So with all this in mind, thinking about how genetic material is recombined during sexual reproduction, what kind of patterns have we noticed? What can we predict about offspring? And this is where the Mendelian genetics comes in. And Gregor Mendel, who's the so-called father of heredity, whose work gave us several laws of inheritance, he couldn't see this. He didn't know what was happening at a cellular level. The process of mitosis and meiosis hadn't been discovered. We didn't have microscopes to see this. We didn't have the technology. It limited our understanding. But what he could see, he painstakingly carried out experiments using pea plants to test what would happen when he crossed plants with different traits. And rather than calling them genes at that point, he called them factors. And actually, there's a fair degree of sort of luck or serendipity there, because he happened to use a species and looked at traits where things were quite clear-cut, all or nothing kind of binary traits a lot of the time, and they were all inherited separately. So there's skill and patience and dedication, and also a bit of luck often involved with scientific discovery. And then Reginald Punnett, he gave us the Punnett Square, the technique of using a grid or table to represent the patterns of inheritance. So looking at the peas, let's take the seeds, like the actual pea that you eat. The seeds can be yellow or green. And Mendel established that one trait was dominant to the other. And in this case, the yellow colour is dominant to green, and that two, what we would say, pure breeding parents, and a pure breeding in a genetic sense means homozygous, so having 2 copies of the allele for that trait, that if there was a pea plant with yellow seeds and a pea plant with green seeds, and they were both pure breeding, if they were crossed together, all of the offspring, so all of the new pea plants in the first generation were uniform. They're all the same. So we know that they're going to be heterozygous, if they're crossed from two pure breeding parents. And if they all appear yellow, that tells us that yellow must have been the dominant allele. So the parents, the convention we use, we would denote P to say the parent generation, and then the first generation bred from that, we use F1. And if we cross the F1 amongst themselves, so all the heterozygotes, they can be bred to make the F2 generation. And if we pop that into Punnett Square, we get a phenotypic ratio of 3:1. So let's have a look what we would use for that. And one convention we can use, which is the oldest one, is to take a single letter of the alphabet to represent a gene. So you would take the letter usually from the dominant trait, so we'll go yellow for a Y, and we put it in uppercase for the dominant yellow. And then the green, we still use the letter Y, but we put it in lowercase to show that it's recessive. So building on this, we have a look over here. That's just your Punnett Square that you're probably familiar with already, or for IB students that you've done at standard level. So you can see the phenotype ratio of 3:1 there, which is not the same as the genotype, but we're just looking at what actually is expressed, what it looks like in terms of being yellow or green. And that was a monohybrid cross. So we can now follow more than one trait at a time. So let's say we took the example of pea plants and we looked at their seed colour still, yellow or green, but also this time we want to look at the flowers. And they can be purple, which is dominant to the recessive white colour. So Mendel, he gave us the law of segregation, and that means that the inheritance of one trait shouldn't affect the inheritance of another. So what that means is it shouldn't matter if a seed is yellow or green, the peas, it should make no difference if that plant has purple flowers or white. You could get theoretically any combination of those two. So the dihybrid cross is the Punnett Square we use. And let's say we've got a pure breeding homozygous parent pea plant that's got yellow seeds and purple flowers. And another plant, pure breeding homozygous, but it's got green seeds and white flowers this time. So we would cross them together, and the F1 generation, that's going to be heterozygous for both. And then the F1 generation, we can breed them amongst themselves and see what happens to follow them through our Punnett Square. Now, let's just go back for a second to our meiosis picture and focus in here on metaphase 1. And imagine the seed colour gene is, say, here, and the flower gene is here. Like, we've got different spots where that information is carried. They're on separate chromosomes. So when the bivalents line up at the equator of the cell, so they're in their homologous pairs, there's no set side. There's no predetermined side for maternal and paternal chromosomes. So what I've drawn here is red and purple. And if we imagine one has come from this pea plant's mother plant and this one from its father, they reproduce sexually just as, well, not just as we do, but genetically speaking, they do. So what I'm saying, though, is those chromosomes in the pairs, one comes from your biological mum, one from your biological dad. And when they all line up in metaphase 1, it's completely random which side of the cell they're on. And we call that random orientation. Like, have a look at those combinations here. We've got 4 possibilities for those 2 chromosomes. So if we want to make a Punnett Square to follow the inheritance of more than one trait at a time, we have to show all those different possible combinations. And we need our grid to be big enough here. And the possible combinations we've got, as we said, we've got 2 alleles, or 2 possibilities for each gene. So we take 2 and we raise it to the power of the number of genes we're considering here. So we end up with 4 possibilities. Or we can see here that big Y could go with big P, big Y could go with little p, little y could go with big P, or little y with little p. So they're all our different gametes we could make from those cells. And since we're breeding F1 plants with other F1 plants, combinations are the same across the top and the bottom. When fertilization occurs, we write the alleles of each gene together here. So the Y's together as a little pair and the P's together. And let's have a look at our combos. I'll try and speed this up a bit. This is a bit tedious. But we can see our grid, we've got 16 outcomes. Are they all different or any of them the same? If I count up the genotypes, some of them are actually double ups because it doesn't matter which way around the alleles are written. So if I list them here, I'll go through and kind of highlight them in blue and then write them down. So, big Y, big P. And then the next one, big Y, big P, little p. Well, it actually doesn't matter. It doesn't matter if you write the big P or the little p first out of that combination. Those genotypes are the same. And again, we've got another genotype we've identified with 2 out of the 16. Big Y, big P, little p, so heterozygous for both of those genes. We actually have 4 ways we can get that in our dihybrid cross, but that's all one genotype. We go through and keep going and so on. Ultimately, what we see here, if I kind of group them together and have a look at what we've got, each line there, so all those different lines written in black, if I count them up, I have 9 possible genotypes. But also if I look at phenotypes then, so what we would see looking at the plants, I'll just pop this couple of squiggly arrows over here. 9 out of the 16 possible combinations would show both of the dominant traits, yellow seeds and purple flowers, because we only need 1 in each pair to have that allele, so it's pretty common. Then these ones are yellow seeds with white flowers. These are green seeds with purple flowers. So 3 each of the Ps that express 1 dominant trait and 1 recessive in our combos here. And that just leaves 1, sorry, I didn't leave myself much room, it's a bit squishy, but 1 that is recessive in both traits, having green seeds and white flowers. And that ratio of phenotypes, 9 to 3 to 3 to 1, is worth remembering because it crops up in exam questions a bit, but it only holds true when you have this scenario with parents that are heterozygous for both traits. That's when we get these ratios. Your dihybrid cross would look very different if you perhaps did a test cross with something that was homozygous recessive. You would get different outcomes. This is only for heterozygous in both traits. And we can see the 9 is both dominants in the phenotype, 1 both recessive, and the 2 3s there for 1 of each. The other thing it does, though, knowing these possible outcomes and looking at these ratios, though, it allows us to also investigate if Mendel's laws are holding true for our example. And that picture of metaphase and indicating those genes were on different chromosomes, that might not actually be the case. What would happen, if the genes were in fact on the same chromosome? And going back to our homologous pair in red and purple up here, the genes A and B that we just kind of invented, they're what we would call a linkage group because they're carried on the same physical piece of chromatin there. They're on that chromosome. They are going to be transported together and they move to an end of the cell. So we would not get the same ratio that we've just looked at in our dihybrid cross. The only way they can get separated, though, as we see in this picture as well, is through crossing over, and that is still pretty common. But if the loci are quite close together, like where those genes are positioned, the chance of them getting separated is usually still smaller. If the loci are quite close together, like where those genes are positioned, the chance of them getting separated is usually still smaller. The ratios we would get would still be different to a dihybrid cross, but you might not be able to tell at first glance. You might have to do a statistical test. So how do we check? What stats do we use? Thomas Hunt Morgan, he won the Nobel Prize in 1933 for his work on fruit flies. Now, bear with me here. This took me a lot longer to draw than I care to admit, but I'm pretty proud of it. My little Drosophila over here. These are the fruit flies, and they're what's called a model organism because they were useful for doing quick, easy, accessible studies that we can use to model what might be happening in our own bodies. Thomas Hunt Morgan, he also looked at sex-linked traits, but we just want to stick with an example without that complicating factor for a minute. So let's say we did an experiment where we had a wild type fly. Now, the term wild type, that just means it's like saying what's most common for the species, or like what would be considered normal, what alleles they would have in the wild. So a wild type fruit fly has kind of red coloured eyes and its body is tan and black. So I've really only got kind of like an orangey colour there with my highlighter. But that's our wild type fly. And our second fly, and I've attempted some sexual dimorphism here. I've made his bum too big, but that's meant to be a boy with his rounded bum. It should be smaller. But our second fly has purple eyes and a plain black body. Now, both of our flies in our little experiment here, pure breeding, remember that means they're both homozygous for their alleles. What makes it a little tricky, there are other genes that affect eye and body colour in fruit flies. And actually, there's some of them, there's a bit of a hierarchy of what's dominant to what else. So another kind of convention we can use for writing down the genes to put in a dihybrid cross, we might give a letter, or sometimes even two, but we might give a letter for the gene locus. Let's say in this time we're going to say P for the purple eyes and B for body colour, or black body if you like. And maybe we don't know which one's dominant yet, or maybe there are other things that aren't in part of this experiment that are higher on the list that can be dominant to both. So we are just going to use lowercase letters, and this time we're going to use other superscript little symbols to tell them apart. And the convention here that's often used in these experiments with Drosophila particularly is to use a little plus. And we use a plus, and that has nothing to do with which one is dominant and recessive. The plus actually just indicates which allele is wild type, the one that's more common in the wild. So we're going to put a little plus here for the red eyes and the tan and black body. So if you get an exam question that uses this notation, you've got to read the rest of the information to work out which allele is more dominant, because that's not what the plus means. It might be by chance, but that's not by definition what it means. So in this scenario, our two flies breed, and we find out that all of the offspring have red eyes and tan and black bodies as their phenotype. So that's how we know that they're the traits or the alleles here that are dominant. So we'll just jot that down so we know the wild type ones happen to be the dominant ones. So we've bred them. We've got heterozygous flies now for both traits, and they're our F1 generation, and we can cross them with each other. Now, if these traits are inherited independently, like our example before with our peas, we would expect to have the same kind of dihybrid cross. We would expect the ratio of phenotypes in the F2 generation to be 9 to 3 to 3 to 1. So I'm just going to try and fast forward through that as quickly as I can to show you that dihybrid cross just for practice. All right, now we might run an experiment and we might breed 20 flies, let's say. I mean, you probably do a lot more than that, but we'll stick with that for now. What we can do is we can use that ratio, that 9 to 3 to 3 to 1, and each one of those numbers, take it as a fraction out of 16, because there's 16 possibilities in our dihybrid cross. And we can multiply the fraction by 20 flies that we bred in our experiment to get the expected values. How many should we get if this dihybrid cross is accurate, if we have 20 flies? So we'll pop them in a table here. Okay, so 9 out of 16 as a fraction multiplied by 20, it'd be roughly 11 that you'd expect to show to both dominant traits. Nearly 4 each are the ones with one dominant, one recessive trait, and only about one fly should have both recessive traits. But in our experiment, what we observed was different. Instead, we got 8 flies with red eyes and tan on their bodies, 4 with red eyes and black bodies, 3 with purple eyes and tan with the black, and 6 with purple eyes and black bodies. Seems kind of different, but is it statistically different? So this is where the chi-square test comes into play. I've got my little table here just to try and help you follow it. First, we find the difference between each observed and expected value. So we subtract expected from observed. Now, those answers here, some of them are negative, some of them positive, depending if the value is smaller or larger. We square them, though, and that gets rid of any negatives. So we square the answers here, and then the next step is to divide those answers by the expected values. So just circle that in yellow to show that like 10.6 over 11.3, and we get an answer there. Then the chi-squared, that formula, observed minus expected squared over expected, and then that sigma, that sum of, say, all of those values added together gives us our chi-squared value at the end. So they add up to 19.2. Now, what does that number mean? Does it mean that we follow our patterns of inheritance, or do we have something else going on? So we have to consult a chart of values. Definitely not something you need to memorize or ever write out. This is a waste of time, but you can look it up, or you will be given it in a question, and you can then find out if your chi-squared value is above or below a critical value from this table. So let's have a look which part you might use. Firstly, you need to select the right degrees of freedom. For contingency tables, you subtract 1 from the number of columns and rows and multiply that together, which you might have used in ecology. Here we just have a list of 4 possible phenotypes. So we subtract 1, and that tells us we should be using 3 degrees of freedom. If you had more phenotypes, if you're doing something like a trihybrid cross, you'd need to have greater degrees of freedom, which would allow for the fact that you'd be adding more values together. So you'd expect your chi-squared value to get bigger if you were involving more genes, because you're just adding more numbers together. So you've got to pick the right row table to use. Look at this one. Now, if our chi-squared value was small, it would be telling us that the difference between our expected and observed values was small. So when there's a little difference, in statistics we call this the null hypothesis. We write H0 for that for short. And if our chi-squared value was small, we could not reject the null hypothesis. We'd be saying that the dihybrid cross was actually maybe pretty accurate. And the alleles, they get segregated in meiosis and sorted independently, so they're on separate chromosomes, and the inheritance of one trait does not affect the other. Our number is big though. In fact, it's bigger than all the values in the whole row, and they get larger as you move across to the right. This tells us that the differences between our observed and expected values are too large to put down to chance. We reject the null hypothesis. We are saying it is too unlikely that these alleles are sorted independently. Actually, there's a good chance they might be linked together. And we can be pretty confident of this, the bigger our value gets. This row of the table tells us the probability associated with what we're saying. See how it gets smaller as we go to the right? This is kind of like saying it's the chance we're actually wrong about our whole chi-squared test. So in our example, since our chi-squared number 19.2 is bigger than even this one, 16.27, then we are effectively saying there's only or less than 0.001 probability, or as a percentage, a 0.1% chance that we are wrong. The other way to say this is that we can be more than 99.9% sure that our experiment really does differ from the dihybrid cross. So we can be pretty sure our genes are linked. They don't always get inherited together though. We did still get some different combinations. Some cases aren't as straightforward as this, because we still get recombinants from crossing over. And in fact, we did here as well. But in this case, not too many. It turns out that the loci of the genes we happen to pick, for our example, are actually really close together on the same chromosome. And if the genes are really close together, the chance of them being separated by crossing over is lower. Lastly, we also need to consider that some traits aren't affected by just one gene. In fact, look at ourselves. Very few of our traits are down to a single gene. But there are some examples, like having a straight hairline or widow's peak, or whether your earlobes are free or attached. And we would call this type of variation discrete, as people fall into categories. It could be represented by a bar graph. Like here, another example is blood type. But what about features like height or skin color? Here we see continuous variation that follows a normal distribution. These traits are influenced by several genes, which we call polygenic inheritance. In fact, research suggests that it could be over 150 genes that have at least some influence, whether direct or indirect, on our skin color. And making our phenotypes even more truly continuous, rather than just lots of close together categories, is also our interaction with the environment. You might have a genotype for quite fair skin, but spend a lot of time in the sun. So we're pretty complex. Not everything can be examined with a Punnett square, But it's nice to appreciate that the scientists who've used model organisms and kept meticulous records looked for patterns, noticed discrepancies, and done their utmost to be objective and avoid confirmation bias where possible. They've paved the way for our understanding of inheritance.